we know that there are many versions of ESM models, right? We have ESM1B, ESM1B, and
Yep.
we have different Cs for ESM1B and also the
Yep.
ESM2 model. And even we have the ESMC model right now. So, so the goal of their model is that, of the paper is that they took all these ESM models and train a single model. For example, the same architecture as a ESM2 model, but the parameter is different. And when we want to use them to use that model for variation effect prediction. But let's think about it. The nature question is that why do we want to use multiple models to predict the variation effect at the first place? So uh, we know that this is from Kurt's modified paper. In Kurt's modified paper, we use an ensemble of five models, which is ESM1V, ESM2, EV mutation, EV, and MSA transformers. We combine the scores of these five models. And for ESM1V, if I remember correctly, we have different five Cs of different ESM1V models. We add them directly, and we get the average score of them, but it's a weighted average. And we use this score to design the lab libraries. But basically, this method is, I would say, it took sequence as input and is a zero-shot fitness prediction. And we found out that the information at each different model is somehow complementary to each other, which means that each model has its own bias. So and I feel like this paper that I'm talking about has the same motivation of modify, but in modify we directly combine the different models. But in this paper, what they do is to distill a new model which contains all the informations uh, of the different ESM models. And the task here we care about is the variation effect prediction. And so specifically, the data sets we have here is the protein gene data set, and we can and they calculate the Spearman correlation here, and also the CleanMar data set, which is a pathogenicity data set, and the metric here is the AUC. And so the other motivation of their method is that they are trying to uh, develop a sequence-only method because I feel like recently there are also some debates on whether the model should be using only sequence as input and using multimodals as input. And their argument is that some of the sorter methods they use MSA as input or structure as input, and for some pathogenicity data, uh, population data has been used for the input. For example, this UK biobank. And their argument is that using different modalities as input can make the model pretty complex. And also the problem is that sometimes these are data biased, which means that sometimes you do not have this extra modality data, and then your model might not perform as that well. And this will lead to the limited applicability. And also the model can be biased because usually we have many sequences, but we don't usually have many structures or this population data. And yeah, but recently they shows that even the sequence only models like ESM2 can perform quite strong on the tasks that they have. And one observations that they have is that different ESM models have already captured different signals in the proteins. For example, they shows that ESM1B model can detect the KRAB domain, which is shown here. This is a KRAB domain, and this heat map is the predicted scores of the ESM1B models and the ESM2 model. And it shows that ESM1B can detect this domain, while ESM2 failed to do so. And we can see that when we say that ESM2 failed to do so, ESM2 gives a really high score to the KRAB domain, which means that they think all these mutations are pretty good. And also sometimes ESM2 can find domains that ESM1B fails to do. So this BRICHOS domain was captured by ESM2, but was failed to captured by ESM1B. Oh, yes, and this is a visualization of the domains that I talk about. And in ESM2, it literally does not give any reasonable or insightful scores to this protein, and the S1B successfully gives some different scores to it. And this suggests that the knowledge is already there, but they are distributed across different models. So instead of building a new model, the question is that can we extract and combine this knowledge? So I will briefly introduce their method. The first step of the method is to collect the predictions from multiple ESM models. And each model here produce a LLR score for the mutation. The LLR score is the delta log likelihood that we talk about. So a lower delta log likelihood here means the mutation is more deleterious. And instead of averaging all these scores, the paper takes the minimum delta log likelihood as the label. So intuitively, if any model strongly believes the mutation is harmful, we will keep the signal, and we trust the most pessimistic experts. And this will give us a strong pseudo label for each mutation. And my comment on this taking the minimum is that if we take a look at this, ESM2 fails to predict this KRAB domain because ESM2 will give a really high score, high LLR score to this. So I guess that's why they will take the minimum instead of the average of the output of the models. And next, they will train a model to match the pseudo labels using the MSE loss. This is a pretty common approach to co-distill the parameters of different models. And I feel like the most important part here is taking a minimum of the outputs. This is different what we did in the modified model and also the SPERSAX model. But the problem is that only using the minimum of the outputs can be pretty noisy. So they are actually using a three-round approach. In the first round, they are training the model on human proteins, and they are taking the minimum of the delta log likelihoods as the pseudo labels. And at the second round, they are using the human proteins, but the pseudo label here becomes the average. And at the third round, they are using the non-human proteins, but still using the average of the delta log likelihoods as a pseudo label. And what they claim is that at the first round, they are trying to let the model to be pretty sure about which mutation is deleterious. And for training stability, they add the round 2 and round 3 to help the model to have a better generalizability. And after the distillation, the single model quickly approach the ensemble performance. You can see that the ensemble, this is the performance of the ensemble, and after 3 rounds, the model performance can reach the ensemble performance. And also their model, which is the blue dots here, can perform pretty well on DMS data, which is the protein gene data, and the ClinVar data set. But the question is that how do they compare to the existing methods? And so they evaluate the model on the ClinVar dataset, and the metric here they're using is the AUROC, and they found out that their model, which is called VESM, performs the best comparing to previous method. And I feel like the best method for protein at ClinVar should be this ESM1B model. This is really famous, and that those guys who are developing the DNA models are comparing with the ESM1B model. And also, I have to mention that this model follows the scaling law. Because I feel like this is really interesting because we know that in the protein gene dataset, the ESM models does not follow the scaling law. ESM 650 million performs the best on the protein gene. And a previous paper is called LFB. I really like this paper and shows that uh, if you're uh, if you're using the average output of the MSAs as the input, then it might help the ESM models to still follow the scaling law here. I will show in this figure A, and as you can see that as the parameters gets larger, this ESM3B model will not follow the scaling law on the protein gene dataset. This dashed line is the original ESM model. And while they apply this LFB, which is also an ensemble method, it still helps the model to uh, uh, follows the scaling law. And interestingly, this VESM model, which is the model name of this Nature Methods paper, that follows the scaling law. So this might also shows that ensemble might help with the model to follow the scaling law and which means that they can overcome the bias of this larger models. And this might be a way to deal with it. And for the other experiments, they shows that, okay, for sequence-only models, their VESM model performs pretty well on this human DMS datasets on protein gene and also on the ClinVar datasets. And shows that on other tasks such as stability and binding versus fitness and activity, they also performed pretty well. And the other experiments which is also really interesting is that at gene level they predicted that shows that their prediction can somehow correlate with the real phenotype associations. Because as you can see, the y-axis here, some of the predictions are actually negative, and some of the predictions are positive. And this actually shows that this model is not just making those correct predictions and might also capture some meaningful biology.